Hi, welcome to Detours, Understanding Acquired Brain Injury. We discussed in the last series about cells, how genes and chromosomes work. Now we're moving into neurogenetics, but in order to get into the weeds, so to speak, about different kinds of neurogenetic disorders, we do need to look at how cells can malfunction, what happens genetically and epigenetically into things that go wrong, problems, as it were. So let's dive right in. From fertilization to death, genes influence strongly, very strongly, not only the growth and development, but the day-to-day operations of cells. And we mentioned that previously, but it's important to know that at any time during a cell's going about its business, things go wrong. And sometimes that is due to a problem which developed shortly after or when the embryo was fertilized, or it can be something it carried forward from either the sperm or the egg. A lot of people will say, well, gee, so-and-so was always healthy or whatever. Not necessarily. Sometimes genetic or chromosomal disorders can take years or decades to show up, even though it was present at the moment the egg was fertilized or before. And so it's important to keep that in mind, because sometimes genetic disorders take a long time to show up. Alzheimer's, whether it's familial or whether it is due to cumulative damage or whatever, is an example. Or cancer. The problems with the genes involved in this may take a really long time to show up. So, just to keep that in mind. We will get definitely back to this later, but just to touch on it. So we will, with these few starting videos in neurogenetics, we'll look at how things go wrong at the chromosomal, genetic, and epigenetic level more generally, and then we will get into the very specifics when it comes to the conditions we talk about. Okay? So first, let's take a look at problems on the chromosomal level. Usually, when there's a problem with chromosomes, it's a problem with the structure of the chromosome. That is either the number of chromosomes or the shape or form of the chromosome. Remember that the chromosomes are carriers of the genetic information, have the genes attached to them, bound to them. But the chromosomes themselves are not carriers of genetic information, or it's kind of like recipes in the book. The book itself can be damaged and therefore affects the recipes that are in the book. So what are some of the chromosomal structural errors? Most commonly for a chromosomal error is something called aneuploidy. And this is a condition in which there are either too many or too few chromosomes in a cell. That's what aneuploidy is. Now remember, there are different chromosomes. There are autosomes, which are the chromosomes associated with the body, and then there are the sex chromosomes. Most people immediately think of the sex chromosomes, X or Y, and, well, there are 22 pairs of other chromosomes. Those 22 pairs of chromosomes are the autosomes. And X or Y, which determine sex and other things too, by the way, are the sex chromosomes. And the X chromosome carries a lot of autosomal information, stuff about the body and the brain. We'll get into that in a moment. So in humans, the proper number of chromosomes in a cell is 46. And we use the term euploid, meaning everything is good, that the correct number of chromosomes being 46, so a cell is euploid if there are 46 chromosomes, or 23 pairs. That's the other way of saying it. If there's a problem and there's too many or too few, we'll say it's aneuploid. So when there's an extra copy of a chromosome, it's usually due to a problem during cytokinesis, when the chromosomes are pulled apart. So a pair of sister chromatids, let's say chromosome 6, for example, in a skin cell, if they remain together and are pulled across, and they end up in one of the two skin cells, which is dividing and not in the other, this should lead to destruction of both cells. The skin cell with a double set of 6, and the one that's missing. They should automatically trigger self-destruct. And these kind of things do happen all the time. And the cells normally, when they're going through those checkpoints we talked about in the last series, normally they'll destroy themselves. Because if they don't, it can lead to cancer. When we get into things like brain cancer or, leukemia or other things which can affect the brain, we will discuss this in much more detail. But it's important to know that aneuploidy is a serious problem when it comes to cancer risks. There are multiple, what are referred to as these checkpoints in cells, which normally prevent these kind of mistakes. Normally throughout the body cells, but occasionally there are those that slip past. It's normally a much bigger problem when an error like that happens to the gametes, the sex cells, sperm or egg cells, because an aneuploid egg or sperm can become a new person, and therefore potentially all of their cells become aneuploid, because you start with the one and it replicates from there. So if it's a sex cell, this can lead to, some very well-known conditions too. Usually these cells will self-destruct, but not all the time. We are more concerned about aneuploidy in these cases because of what they can lead to and how they can affect someone. Now, the most common kind of aneuploidy is called trisomy. And that is where, in sex cells, where you will get, other cells too will have this, but we're going to be focused more on sex cells. Trisomy occurs when there's an extra copy of a chromosome, one extra copy. And depending on which chromosome, it typically leads to non-survivable conditions. A survivable, but a serious neurodevelopmental disorder with life-shortening expectations or conditions that are noticed by, or not noticed by anyone can occur as well. But trisomy typically can be caused by what's called a non-disjunction. Non-disjunction occurs when the daughter chromosomes, remember from the last video series, when the daughter chromosomes fail to separate and they're still bound together in the middle there, and both are pulled into a cell. So non-disjunction, and you get two instead of the one, so that the paternal, and typically this has to do on the maternal side, where instead of going into the egg, just one, so that the paternal chromosome can fertilize. Instead, you've got two already, and then you get third. And that's where the trisomy comes from. Now, there are a bunch of different trisomies, but unfortunately, most are not survivable. There are only a couple kinds of trisomies that are. So this 2+1 pattern. The survivable trisomies are trisomy 13, so that is called Pateau syndrome. Looks like this. This is the effects of it. It's very, very serious, and usually, even those who survive, who survive birth, don't live very long because it affects internal symmetry as well as external symmetry. Affects the heart, the brain, digestion. It affects. This is probably the most lethal of the survivable asymmetries, or the trisomies, with apologies, of the trisomies. Trisomy 18. It's called Edwards syndrome. Again, this is usually not, although we're getting better with it. This one is very condition where someone's very unlikely to survive. This girl probably has mosaic Edwards syndrome, meaning not all of her cells have the trisomy, and that happens later in the process oftentimes, where this kind of error, and we will get to mosaicism. And of course, the most famous one, and we're doing a lot better now with treatment, and kids are very likely to survive with trisomy 21, Down syndrome. And it is an extra 21st chromosome. 21st chromosome is the shortest chromosome. It has the fewest extra genes, and therefore it is most survivable. But those are trisomy 16 is survivable if it's mosaic. If it's a complete trisomy, not so much so. Or partial trisomy 16, survivable. But that seems to be it. Most trisomies miscarry if they're for other chromosomes, just because the critical genes on those, if there are three chromosome pair, or if there are three chromosomes instead of the pair, it leads to lethal errors, and it tends to miscarry. And any embryo with this disorder, with other autosomes in trisomy, and that's the other thing. This applies to autosomes. Let's look at the sex chromosomes in a trisomy. Remember, these aneuploidies are extras or deletions. All right, so the trisomies of the sex chromosomes include things like 3Xs. It's just called triple X syndrome. No fancy, no person's name, because it wasn't discovered years ago. This is a more recent discovery because its effects are kind of non-specific and confused with many other disorders. Klinefelter syndrome is 2 X's and a Y. 1 Y, 2 X's. And it's again, there's fairly non-specific, but there's oftentimes like ambiguous genitalia. There are a lot of sometimes cancers and other problems that are, we will talk about it in detail later, but there are neurological issues, things like that. But this is a non-specific one. Then there's Jacob syndrome, which is an X with 2 Ys. Back in the 1960s, 70s, when they discovered this, they assumed, super criminals and all this kind of stuff. No, not true, not at all. They just assumed 2 Ys made you. The SRY gene, which codes for, male traits, only one is readable. The second one is not. It more likely to lead to cancer, again, more likely to lead to neurological problems and cardiac, bone problems. It's just an unfortunate situation. There are no cases of a bunch of Ys and no X. That is non-survivable. I've mentioned this before, but you must have an X, at least one, and we'll get into that kind of aneuploidy in a moment. But other sex-related aneuploidies, where you have duplications, you can have 4, 5, or even 6 X's, but the effects on the developing embryo are more and more serious, and intellectual impairment and all kinds of other motor problems and developmental problems become so serious that eventually you hit that non-survivable point again. And that's the thing to remember, is that most of these are non-survivable. Very few. Even in mosaic cases or partial cases, because it produces extra genes with extra proteins that interfere with the function of the cell. All right, let's look at the other kind of aneuploidy, which is deletion of a chromosome. And again, we're talking about complete. We're not yet getting into the partial, but it's critical. There is only one survivable complete chromosomal deletion. One. That is called Turner syndrome, and that is a sex chromosome deletion. And that is where a female has an X, and she does not have another X or Y. And that is because another egg cell has given 2 X's, and there's no X. So she either gets an X from her father or would get a Y if she were male, and that Y without anything is non-survivable. So in cases of Turner, there is an X, just one, which poses a set of other problems. But you would describe this as an X. Second, you would just say there's an X chromosome and nothing, XO. Turner syndrome. We'll get into this because this is a neurological disorder, commonly. Specific visuospatial problems, learning disabilities in the US sense, not the British sense of MR, intellectual disabilities, but learning with like nonverbal and that kind of thing, but also some motor deficits and things like that. Again, it is the only survivable chromosomal deletion, complete deletion. Partials and things like that are survivable. So this ends the first section of neurogenetics, and the most dramatic kind of problems you'll see in chromosomal disorders, which is aneuploidy, which is the complete doubling, tripling of both autosomal and sex chromosomes, as well as deletion, full deletion. The next chapter, then, we will discuss some of the other things that can happen with chromosomes, which are dramatic but don't affect quite in the same fashion. All right? So, hopefully this was helpful and you now understand some of these disorders. We'll move on in our next unit. Please, as always, click like and share. Share this with anyone who may benefit from understanding, and we'll see you next time. Bye.